Well, great. I would really like to thank Dr. Reed and the other organizers for inviting me to participate in this microbiome course. I have no disclosures. I was asked to talk about the microbiome of the urinary tract in pediatric patients, but as was very nicely discussed by Dr. Vasquez, you will see that I can't really discuss the urinary microbiome without discussing the microbiome of other surrounding organ systems, as they significantly impact the urinary tract. As an outline of my talk, I will briefly set the context of urinary tract infections. I'll discuss the limitations of using antibiotics in their treatment and prevention, and then the roles of a variety of microbiomes on urinary tract health, hopefully without repeating too much of what Dr. Vasquez has already said. And lastly, I'll go over some data on probiotic use in UTIs. We all know urinary tract infections are one of the most common outpatient bacterial infections, accounting for 150 million cases worldwide each year. In the US, pediatric UTIs affect nearly 3.5% of children annually, with a recurrence rate of 12 to 30%. UTIs in children can have significant long-term sequelae, including an increasing risk of renal scarring with number of febrile infections. And these facts highlight the necessity of understanding risk factors for infections and finding means to limit UTI susceptibility. There are some critical steps that enable microbial access to and growth in the urinary tract. First is bacterial ascent from the perineum. We do know that the uropathogenic bacteria arising is usually arising from the GI tract. The second is bacterial adherence to glycoprotein receptors on the uroepithelial cells via bacterial structures called pili. While antibiotics are the mainstay of therapy with pretty universal acceptance of their use during acute infection, their use to prevent infection is more controversial in the pediatric population, with certain seminal studies showing prophylaxis decreases urinary tract infections, such as in the River and Prevent study. But a 2019 Cochrane database systematic review did not show any benefit of antibiotic prophylaxis. In addition, there are risks with using antibiotics. They have side effects, accounting for not an insignificant number of emergency room visits per year in the United States. There are also concerns regarding rising bacterial antibiotic resistance rates, linking rising patterns of resistance to antibiotic use. A systematic review found an increased risk of multi-drug resistant urinary tract infections in children with vesicoureteral reflux on antibiotic prophylaxis. This has led to a push for real antibiotic stewardship and trying to limit antibiotic use and an interest in exploring antibiotic independent means of preventing infection. An important concept related to UTI susceptibility is knowing that our bodies are comprised of a host of bacteria at baseline. Dr. Vasquez very nicely outlined this, but really the bacteria cells outnumber our human cell numbers by 10 to 1, with this schematic giving you a sense that our own cells only comprise part of a leg relative to the other cells to which our body plays host. As a result, specific microbial signatures exist in our body that are involved in the maintenance of health and susceptibility to disease. Our understanding of the human microbiome is predominantly a result of the Human Microbiome Project. This Human Microbiome Project is an extension of the Human Genome Project that began in 2008 with a goal of characterizing the bacterial makeup of various body sites. It is the first large-scale mapping of the human microbiome using culture-independent methods, with really the project being made possible due to the emergence of molecular methods to study microbial communities. And these methods significantly expanded our ability to identify different microbiota. Early characterization of microbiota relied on culturing, but we now know that a significant amount of microbiota in at least the GI tract and oral cavity are unculturable. There have been some advances in culture techniques, such as enhanced quantitative urine culture, that can identify bacteria beyond that grown in standard culture. But newer molecular methods of diagnosis and identification of bacteria really significantly advance the field of studying the microbiome. This includes targeted amplicon study approaches, such as 16S rRNA next-generation sequencing, and the large-scale metagenomics approach, or shotgun sequencing. The different microbiomes involved in urinary tract health include that of the gut, vagina, perineal or periurethral, and urine. Each site harbors diverse ecosystems of microbes that regulate interactions with the local environment, and dysbiosis results in disease states. There is an interrelation between sites, with these sites in particular under the influence of one another, largely due to their proximity and the potential for cross-contamination. There is an ever-expanding literature on the gut microbiome, with it being the most readily studied microbiome at this time and with the most impact on other organ systems, with an association of early life imbalances of the gut microbiota with several diseases, and again, was really nicely outlined by Dr. Vasquez. The development of the intestinal microbiome begins at birth, with age being the major driver of intestinal microbiome changes. The diversity of the microbiome nearly doubles in the first year of life, with the first 3 years of life being the time of most change in the intestinal microbiome, which tracks with behaviors during the first 3 years of life that increase the exposure of children to microbes, such as breastfeeding, sucking on one's hands, crawling on the floor, and transitioning to different types of foods. By 3 years of age, the intestinal microbiome is thought to be more stable and representative of an adult-like gut microbiota. There's a strong relationship between the gut microbiome and urinary tract health, as we previously heard. We know the fecal flora is the origin of most of the bacteria causing infections, such as E. coli. The GI tract is the primary reservoir for uropathogens, with an association found in adults between increased periurethral carriage and fecal load of uropathogenic E. coli immediately preceding an infection. There is a gut microbiome signature that is associated with UTI risk in the pediatric population, with a study demonstrating differences in bacterial family in children with UTI versus controls. Mutations in E. coli virulence factors were also found more often in those with a UTI compared to healthy individuals, suggesting the presence of more virulent uropathogens in the gut of those with UTIs. There is a concept of colonization resistance, in which local bacteria in the GI tract is thought to inhibit colonization by potentially pathogenic microorganisms. Given the majority of uropathogens arise from the microbiota of one's own GI tract, methods to prevent ascending spread of bacteria from this ecosystem to the urinary tract have focused on boosting the surrounding healthy microbiota via probiotics and altering the composition of potential uropathogens present. Dr. Vasquez mentioned the vaginal flora, and we know this plays an important role in urogenital health. The predominant commensal organism is Lactobacillus species. And these species of bacteria can be found in the vaginal and periurethral areas of healthy premenopausal women and play a key role in urinary tract health. Reductions in vaginal lactobacilli is associated with increased vaginal E. coli in adult women, with depletion occurring after sexual intercourse, spermicide use, and menopause. Interestingly, all known risk factors for infections. Repeated antibiotic use also adversely affects the local lactobacilli composition of the vagina. Lactobacilli work, as mentioned, by several protective mechanisms. They block uropathogen adherence, produce antimicrobial substances, compete for nutrients, and modulate the host immune system. There is variability in lactobacillus strains, as was mentioned before as well, with some working through certain mechanisms more than others. These differences are important when assessing their potential role as therapeutic agents to prevent infections. While there's a fair amount of data regarding adult female vaginal microbiota, there are fewer studies of the vaginal microbiome in healthy children, and specifically regarding age-related changes. The prior work that was done was predominantly culture-based. There has been some question regarding the maturation of the vaginal microbiome and the acquisition of lactobacilli. It has been taken for granted that the female flora, and particularly lactobacilli, is acquired with puberty, partly due to findings of loss of lactobacilli species with menopause and its association with estrogen levels. A prior study of healthy females using culture data found that lactobacillus was present in older compared to younger girls. A more recent study was performed showing that lactobacillus can be isolated prior to puberty. In this prospective longitudinal study of perimenarchal girls, in which investigators took vaginal swabs over 3 years, they found that lactobacillus dominated the vaginal microbiome even before the onset of menarche. Using next-generation sequencing, they found a predominance of lactobacilli in the vaginal samples at all ages, including premenarchal girls. Overall, this study suggests that the vaginal microbiota of girls begins to resemble that of adults before menarche. I was involved in a study, as Dr. Reed alluded to characterize the evolution of the perineal microbiome of healthy female pediatric patients, particularly at different developmental milestones. In this cross-sectional study, we collected 4 swabs of female patients at specific developmental milestones during childhood, starting with newborns, infants, toddlers, and prepubescent girls that were really premenarcheal. We collected swabs of the periurethral perivaginal area, a perirectal swab, a periauricular swab, and an oral swab. We found statistically significant different populations for community membership for all 4 sites represented by the different colors, with each developmental milestone, indicating that the microbiota is site-specific for each developmental group. Looking at the changes with age, the biggest net changes of the perivaginal, periurethral, and perirectal species was between 6 months and toddler age, and could be related to an evolving diet and local environmental changes, such as transitioning out of diapers. Looking at the presence of lactobacillus, we found higher rates of lactobacillus in newborns and in our premenstrual group, which confirms the presence of lactobacillus premenarche, but does suggest a fluctuation in abundance with age. We did have a separate subgroup of girls who had a history of an infection to whom we age matched to some of our healthy controls. And we compared the perivaginal, periurethral, and perirectal flora of the 2 groups, finding significant differences in the microbiomes between those without a history of infection, noted in blue, compared to those with a history of infection, noted in orange. When looking at the actual microbiotal changes, there were more changes in that of the perivaginal, periurethral than perirectal microbiota. In those females who were UTI naive, so our controls, they had more bifidobacterium and lactobacillus, except for lactobacillus reuteri, which was found more in those with recurrent urinary tract infections. Lactobacillus reuteri is commonly found in oral probiotics, and we did not ask families about probiotic use to know if this could be causing this finding. Similarly, we found more lactobacillus in the perirectal swabs of those with a history of UTI, or I'm sorry, without a history of UTI in the naive group. There is a long-standing belief that the urinary tract was sterile until about 15 years ago. This led to the urinary tract not actually initially included as a site in the study of the Human Microbiome Project. However, some seminal studies using metagenomics showed that the urine of healthy individuals is not sterile. This led to identification of the urobiome, a microbial ecosystem present within the bladder that can be measured within the urine. There are some generalizations of major taxa identified in the adult urobiome, but there's high variability amongst individuals with no core microbial community yet identified. Adult studies have found an apparent evolution of the urobiome with age, and pediatric studies have found the urinary microbiota present even in neonates, although its origin is unclear. It is hypothesized that the main origins of the urinary microbiota are microbial communities of the GI tract and vagina. The investigation of the urogenital microbiome in children is really just starting, with limited work in children so far, but it is known that they do have a recognizable urobiome. So far, 200 different genera have been found in the urine of children thus far by next generation sequencing. Organisms identified in the urinary tract in pediatric patients depends on age, sex, and how the sample is collected. In a study of children who are less than 4 years of age being evaluated in the emergency department for fever with catheterized urine samples, they found the most abundant genera of bacteria to be Prevotella, Peptonipholis, Escherichia, Villanella, and Finigoldia. They found no difference in alpha diversity, which is the number of species in a single site based on age or gender. They did find a difference in the type of bacteria between boys and girls, with boys having Actinobacter phylum and girls having a correlation between the genus Mobiluncus with increasing female age. In another study of older toilet-trained children with voided samples, the most abundant urobiome genera were Poropheromonas, Prevotella, Ezacula, Campylobacter, and Dialaster. They also found the microbiota differed by gender. These studies go to show that the healthy bladders of children display a distinct microbiome to those of adults. There is a growing literature documenting differences in the urobiome and urologic conditions, with dysbiosis of the urobiome related to urinary tract infections, nephrolithiasis, lower urinary tract dysfunction such as overactivity, incontinence, and even neoplasia. The urobiome microbial composition and diversity is related to the development of urinary tract infections, with adult studies showing an association between decreased microbial diversity and the incidence of UTI. Commensal bacteria of the urinary tract are believed to limit uropathogens with an imbalance of urinary microbiota, enabling overgrowth of pathogenic bacteria or even leading to resident bacteria revealing their uropathogenicity. Children have been found to have less diversity of their urobiome in the setting of infection. In the study of children less than 4 years of old being evaluated in the ED for fever with catheterized urine samples previously discussed, they found a difference not only in the alpha diversity, which is the number of species in a single site, but also the beta diversity, which is the type of species between sites, in those with the UTI versus those without, with this figure representing those differences with those having a UTI in yellow and those without in blue. Similarly, they found a decrease in alpha diversity if the patient had been treated with antibiotics in the last 2 weeks of sample collection, with otherwise a rather rapid recovery with return of the urinary microbiome by 1 month after treatment. An interesting recent study out of Boston Children's looked at the relationship of the urinary microbiome in patients with vesicoureteral reflux. The clinical course of children with reflux is unclear. It's unclear which children with reflux will develop a febrile infection and subsequent renal scarring. And this study was looking at the urinary microbiota and metabolome in pediatric reflux. They found different microbial community compositions in those with recurrent febrile urinary tract infections and those without. They also found a distinct bacterial signature in those with reflux and scarring. And they found altered urinary metabolites showing changes in glutamate, tryptophan, and histidine degradation pathways, which are associated with bacterial metabolism and modulation of the host inflammatory response. All this suggests that the urine metabolic signature holds a potential for identifying children with reflux who are at risk for infections and subsequent renal scarring. As almost a planted question about the urobiome and the neurogenic population, I do have some just brief studies to review that have looked at the urobiome in this particular population, with finding that there are alterations in both adults and children. In adults, the urobiome of patients with neurogenic bladder are more commonly contain bacteria typically considered uropathogens. In a cross-sectional study of children, they similarly found bacteria in the urinary microbiome to that of adults, with a difference in alpha diversity between those with and without a positive urine culture, but no overall difference between those with urinary tract infections versus asymptomatic bacteriuria. And this was felt to be related more to the limitations, as was alluded to during the question and answer session, in the definitions used to diagnose urinary tract infections versus asymptomatic bacteria, and a small patient number in the study. Strategies of manipulating and reshaping disease-prone microbiomes may serve as an alternative or supportive option in the management of pediatric urologic diseases. Probiotics, as we know, are live microbial organisms that confer a health benefit on the host. The most commonly studied are strains of lactic acid bacteria, with those found to be beneficial in managing UTIs, being in the Lactobacillus and Bifidobacterium genera. They can be administered orally, vaginally, or via intravesical instillation. Vaginal lactobacillus is really only studied in adults, with Lactobacillus rhamnosus and fermentum and crispatus showing benefit, while intravesical studies of Lactobacillus rhamnosus in the neurogenic bladder population have been shown to be safe and well tolerated. There have been mixed results in oral probiotics to prevent urinary tract infections in children. In children with urinary tract abnormalities, such as reflux and a history of urinary tract infection, there are 2 randomized trials showing that probiotics versus antibiotic prophylaxis have equivalent rates of UTI recurrence. Specifically, they looked at using Lactobacillus acidophilus versus trimethoprim sulfamethoxazole. In another study, combining the probiotic Lactobacillus acidophilus and Bifidobacterium lactis with the prophylactic antibiotic nitrofurantoin, they found the combination decreased the rate of febrile urinary tract infection compared to antibiotic alone. Importantly, in all of these studies, the use of a probiotic lowered the antibiotic resistance rates of the bacteria that was cultured. In studies of probiotic use in healthy children with presumed normal urinary tracts, there were 2 large studies that found that probiotics significantly lowered urinary tract infection rates compared to no intervention. In a study of girls with past urinary tract infection on the probiotic Lactobacillus rhamnosus compared to no intervention, they found no difference rate in subsequent urinary tract infection. In a systematic review, probiotics are shown to have promise in kids being more effective than placebo in reducing recurrent urinary tract infections in normal anatomy children and as effective as antibiotic prophylaxis in children with reflux. They also lowered the risk of antimicrobial resistance in bacteria compared to those children that were on antibiotics. And a combined therapy of probiotics with antibiotics was the most effective. Still yet, neither the US Food and Drug Association nor the European Food Safety Authority have yet to approve any probiotic for treatment or prevention of disease. So in conclusion, we see that there are unique microbiomes in the human body that contribute to urinary tract health, with personalization of the microbiomes with age, and that children with urinary tract infections have dysbiosis of these microbiomes with factors that predispose to infections and causing their remodeling being unclear. The identification of signatures that increase UTI susceptibility and their sequelae will improve care and promote personalized medicine. And modification of the microbiome alone or in conjunction with other therapies, particularly antibiotic use, does show promise. And I'd be happy to take any questions. And again, I am honored to have been asked to participate in this seminar.